Good morning, everybody. We are the GNN-TOX team, and today we're going to present to you our product. So the mission statement that we have is that we want to leverage graph neural network technologies to reduce the cost and lead time of drug development in order to provide more affordable drugs and reduce the time patients have to wait for treatments. The team members are myself, Julio Soldevilla, and Ryan Kong. And a little bit more background in the problem. So as a drug development usually takes more than 10 years, and it's a problem that has a very low return on investment for big pharmaceutical companies, particularly because it's a very slow process, it's a very long process, and every single step of the process has a lot of inefficiencies. And meanwhile these drugs are being developed, there's a lot of patients that receive suboptimal treatment because there's not the right drug for their diseases. So we would like to avoid this. A few of the questions that we were asking ourselves when we were starting this project were, is there a way to accelerate the drug development process? Can we focus on a specific portion of the process and innovate how things are done? And while looking for solutions to this, we stumbled upon the problem of toxicity. So toxicity testing is one of the most time-consuming tasks in the whole process and causes several candidate rejections. So we figured that speeding up toxicity could speed up the whole development process. So what's the market need for this? Well, on average, the time for a molecule to go from discovery to launch is from 10 to 12 years. On top of this, from 10,000 molecules that are initially screened in the drug development process, only 10 of them ever make it to clinical trials, which is 0.1% of the candidates. And even more, the chance of success for a compound entering phase 1 trials is slightly under 10% and has not increased in a decade. So, additionally, toxicity accounts for more than 30% of rejections, which means that if we are able to catch toxicity early, big pharmaceutical companies could save lots of money by not testing the candidates that are very highly probable to be toxic. So, big pharmaceutical companies could definitely benefit from a process that speeds up the testing of candidates at each part of the process, in particular toxicity. What's the economics behind all of this? Well, as of 2018, average cost of research and development for big pharmaceutical companies is around $2.168 billion. And the average forecast peak sales for a late-stage drug is around $407 million. So as we can see, there's a very, very low return on investment. And the cost to develop and test each of the candidates is of around $1,000 per gram for each of the candidate molecules. So with our technology, we can prevent companies from developing 30% of the drugs that might end up rejected, thus saving the company at least $5 million by predicting toxicity of drugs. So given these numbers, we believe that an approachable price for our service would be $100,000 for a yearly service, which would go directly to our engineers and developers for maintaining our product. So here's just a short graph of the cost of developing a drug in different fields of medicine. So in oncology and immunology, it's the most expensive one, with costs rounding around $2,700 million for developing a drug. Or even dermatology, it's also very expensive, $747 million for developing a drug. So the product that we are proposing is a software that demonstrates potential to screen out promising molecules before any experiment is conducted. This could lead to a significantly reduction in cost and time for development. Our product aims to use state-of-the-art graph neural networks to exploit not only the features from the atoms that make up a molecule, but also their structure, their graphical architecture. And the most important feature of our product will be to have a high classification accuracy, because there are so many different ways that a molecule could be toxic to our body. The data that we're using is the TOX21 dataset. It comes from a challenge in 2014. The dataset contains information on how 10,000 different compounds interact with 12 different pathway assays. And the compounds are represented using a representation of molecules called SMILES, which is a vector of 1,024 elements. And the data is accessed through a Python package called deepchem. I'm going to mention the baseline model performance in a bit, but I want to talk more about the architecture. So basically, our training data is small enough that we can store it locally, and then to develop we used Google Colab notebooks, Python, scikit-learn, GitHub, obviously deepchem, We packaged the whole developed code into a Docker container and used MLflow to orchestrate the end-to-end process of prediction. We stored our trained models in pickled files, which are available in Docker Hub, and then using AWS EC2 instances, we serve our model by surfacing an API endpoint that the client will be able to reach whenever they want the prediction of some molecule. So we did have some alpha release, but we're going to focus on the results of this alpha release. Based on our results, we concluded that we needed to find a model that gives a better performance at the baseline models in terms of the area under the curve, which is because we have a multi-classification problem. And we were facing some issues in the way we uploaded the data to our pipeline, which was causing some bugs in the whole process. And additionally, we found out that our user interface was not very friendly, so we needed to work on making our interface a bit more friendly. The final solution that we came up with, or at least that we had time for fixing, we decided to use a graph convolutional network, which basically considers atoms as nodes and the bonds as edges. The basic idea of such a model is to start with a feature vector for a central atom contains information about this atom, and then we're gonna update this feature vector by somehow combining it with the feature vectors of neighboring atoms. In this case, we're just adding the feature vectors, but we could do the average of the feature vectors, or we could do something more fancy with other techniques. This process is repeated for each of the neighbors of an atom up to a certain radius from the starting point. So here we can see some of the results. The convolutional graph network has an area under the curve of 74%, whereas random forest and SVM have an area under the curve of 55 and 58% respectively. So we can see a huge improvement from just using graph neural networks. For our data set, we have 750 molecules, and the pipeline is running at an average of 3.5 seconds. And so with this speed, we could test around 10,000 molecules in under 1 minute, which is fast enough to start giving some useful predictions to big pharmaceutical companies. So what's our future work? So in order to make our product makes it to market, we need to have a much higher AUC score, optimally higher than 90%. But for that, we still need to explore more architectures and keep doing more tests. Right now, our interface is working, but we can still work on the user experience side of things. We invite you to visit our GitHub page, where we have a repo with all the instructions to run the demo that we're going to present in a minute. Also, feel free to read the paper that inspired the graph convolutional network that we're using. And we are linking or showing some documents on the business side of things for drug development. So with that, please enjoy the demo.
Hello, I'm Ryan. I'd like to welcome you to the GNN-TOX product demonstration. In this video, I'll just go over the basic functionality of our product. So I wanted to start by going over a couple things. First of all, the input to our model is representations of molecules through the SMILES format, or Simplified Molecular Input Line Entry System format. And this is just a standard way of representing molecular structures as strings. So to access the model, we're going to upload a file containing these SMILES encodings, and then we'll be able to download a process file which contains all the predictions for each molecule. Right now, the model currently lives on AWS, and the address is shown here. So let's get to it. If we open up the address, we can choose a file. I can say a test input file. And all this input is a text file where each line is the SMILES encoding of a different molecule. So after I say upload, model inference is instantly run, and then I'm prompted to save a file. So I'll just call this output. And then if we open up the output file, you can see it's a CSV with a fairly straightforward layout. We have the SMILES encoding for the molecule, and then for each of the 12 different pathways of interest in the TOX21 dataset, we have the prediction for whether it's going to be active as a 1 or whether it's not going to be active as a 0. So that's it. It's very straightforward and easy to use. Once again, Thanks for watching this video. For those who are interested, our code is available on GitHub, and our app is also published on Docker as a fully working container. All right. Thanks for watching.